The following podcast is taken from a webinar presented by Dr. Lance Baumgard from Iowa State University titled The Benefits of Mitigating Heat Stress in Dairy Cows. To view the full webinar and access the slides referenced during this podcast, visit balchem.com/realscience and use the search bar to jump to this webinar from May 12th of 2020.
Okay, so this may seem strange to start off a heat abatement talk about the gut, but I hope you'll follow along with me. We'll get to the gut here eventually, but the idea that an unhealthy intestine or an unhealthy gut is the epicenter of an animal's overall health is not new, right? So you'll see in a second how we whittle our way down to the gut during heat stress. But like I said, if you ever experience any type of gut problems, and a huge percentage of the adult population do, you'll understand why. If the gut's not healthy, the animal's not healthy. Just a quick reminder of what the gut is. The GI tract, the gastrointestinal tract, is a tube running from the mouth to the anus. And technically, everything inside that tube remains outside of the body. So that means the breakfast you just had a few hours ago, most of it is remaining outside of your body. It takes a couple hours, 2 or 3 hours before those nutrients are absorbed. It's not until the animal the food stuff is absorbed that actually becomes part of you, right? So, and we as nutritionists and veterinarians oftentimes think of the GI tract primary job is to digest and absorb valuable nutrients, and clearly that's an important job. But if you're a strict immunologist, you'd probably argue that a more important job of the GI tract is to keep unwanted molecules from entering or infiltrating into the body. Most of the things inside of a person's GI tract or an animal's GI tract, they don't want in, right? Parasites, pathogens, enzymes, acids, etc. If these things infiltrate the barrier of the gut, they have an opportunity to stimulate the immune system. And here's just a cartoon of what the villi structures look like. Of course, these villi, their intent is to markedly maximize the surface area of the gut. And there might be thousands of different cells that are lining this particular villi, okay? And because of this strategy the animals use to maximize the surface area of its gut, it really does increase the size of the surface area. And two other epithelia that all animals have are the skin. And I'm just using the skin as a reference because I think everyone has a good idea of how much surface area of skin there is. This is a human example now. There's about 2 meters squared of surface area in a human skin. The lungs, because of the alveoli of the lungs, there's substantially more surface area of the lungs compared to the skin. Because of these modifications in the structure of the gut, the mucosal folds, the villi, and the microvilli, the surface area of a human's GI tract is about 150 times the size of the skin. Right? That's an enormous increase in surface area. Now, this is, again, a human example. So you can imagine then how much larger the surface area of the gut, of a ruminant's gut is. Right? Because obviously the pre-gastric fermentation compartments, this number might be 10,000 times the size of the skin surface of a cow. I don't know, but regardless, it's an enormous number. And again, if you're looking at a human's GI tract, if you laid down the GI tract and flatten its surface area out, it would be about the size of a doubles tennis court. So remember, all those, there's the pathogens, antigens, etc. that are in the gut. In fact, you have more microbes in your gut than you do cells of your body. So, the old joke about who's really the host and who's really the parasite here is clear, right? And because of all these microbes, even the non-pathogenic ones, if they infiltrate into the body, they're going to stimulate an immune response. So it's not surprising then that a majority of the immune system evolved from the gut. Even in the most simplest creatures, their immune system mostly resides in their gut. And still today, over 75% of an animal's immune system resides in the gut. It's because it's massive, right? And it's constantly exposed to potential pathogens.
New research is changing everything we thought we knew about choline's impact on the cow and her calf. and top scientists have a lot to say about it. They are presenting new research that supports choline as a required nutrient to optimize milk production, choline as a required nutrient to support a healthy transition, choline as a required nutrient to improve calf health and growth, and choline as a required nutrient to increase colostrum quantity. This new research is solidifying choline's role as a required nutrient for essentially every cow, regardless of health status, milk production level, or body condition score. Learn more about the science that is changing the game and the choline source that is making it happen. Reassure precision-release choline from Balchem. Visit balchem.com/scientistsay to learn more.
Now, my group, today we're going to be talking about how heat stress initiates leaky gut and the consequences of that. We've since then extended the biology of leaky gut to looking at the transition period and feed restriction and hindgut acidosis. And we're just now starting to look at mycotoxins. But we're not going to talk about these other insults to the gut. We're also very interested in calcium metabolism, but we're going to focus in today on how heat stress initiates leaky gut and how that leaky gut then influences the immune system, metabolism, and the hormonal system, and ultimately how it reduces productivity. A very quick review, though, of metabolism. An animal that's on an ad libitum feed intake and gaining weight. What's from a dairy perspective? Let's use the cow that's 200 days in milk as our example. She's in a positive energy balance. She's ad libitum fed. Insulin levels would be high. Okay, that's the hormone that's reflective to feed intake. And because of the hyperinsulinemia, there's a decrease in non-esterified fatty acids, and so she's putting on condition. Insulin is very potent lipogenic hormone and very potent anti-lipolytic hormone. So she's gaining weight, she's gaining condition, and the hormone that's primarily responsible for that is insulin. Now let's compare her to the animal that's suboptimal feed intake. For example, the cow that's the 10 days in milk. Okay, she's only a week after calving. She has the lowest levels of insulin in her entire lifetime will be during that short window of time. It is this hypoinsulinemia that allows her to mobilize adipose tissue, non-esterified fatty acids go up. So she's losing body condition, she's losing body weight primarily because of the decrease in insulin. Okay, so this is normal metabolism that I'm sure we all had to memorize back in nutrition class. And the reason I'm emphasizing this is because you'll see how different this is when we're talking about heat stress. Okay, so also just a very quick reminder of how important glucose is, especially to a lactating ruminant. So glucose is primarily made from propionate. Of course, propionate is one of the volatile fatty acids coming out of the liver. So this monosaccharide, glucose, is the precursor to lactose. So this is milk sugar. This is a disaccharide. And the reason why this is important is because the amount of lactose made primarily determines the overall milk yield. In other words, lactose is the primary osmotic regulator of milk yield. So the more lactose an animal makes, the more milk she'll provide. And we know that it takes about 72 grams of glucose to make 1 kilogram of milk. So if anything disrupts this pathway, either back here in the rumen, or if anything reprioritizes the use of glucose other than the mammary gland, it has the opportunity to cause milk production. Okay, so obviously we're all interested in heat stress for a variety of reasons, animal welfare being key, but the other reason, of course, is economics. And the dairy industry is very sensitive to heat stress, most of all the animal agriculture industries, at about $1.7 billion a year. Globally, I've read estimates that's up to $500 billion a year. It's hard to wrap your head around, and even hard to calculate, obviously. It'll get worse in the future, so it's already a huge problem. I want to emphasize that. It's already a massive economic problem. It'll get worse in the future if climate change continues. Even if climate change is not going on, even if climate change doesn't continue, heat stress will become more of an issue in the future because of how what our genetic selection continues to emphasize. All of the phenotypes that are economically important to us in animal agriculture, milk synthesis, lean tissue accretion, piglets per sow, etc. These are all heat-producing processes. So the amount of heat an animal produces today, of our farm animals today, is substantially more than what it was 40 years ago. And the likelihood of our grandchildren's farm animals producing more heat than our current animals are is almost certain. So in other words, the temperature at which a high producing animal becomes stressed or heat stress is going to continue to go down. So we have to figure out what's going on and try to understand the biology of heat stress so we can implement mitigation strategies. So one of the first experiments that Rob and I did at Arizona looking at heat stress was how much of the reduction in feed intake explains the reduction in milk yield. So in other words, all animals have a reduction in feed intake when they become heat stressed, right? And forever, we've always assumed that the reduction in feed intake explains why there's a drag in productivity during the summer. So we implemented what's called a pair feeding experimental design. We heat stress cows. We record how much of a reduction in feed intake they have, and then we pair feed a group of thermoneutral cows. So in other words, these cows are remaining at 18 degrees centigrade, 68 degrees Fahrenheit, and we only allow them to consume the same quantity of feed that the heat stress cows were consuming. So they're paired, right? They're paired to the heat stress. Okay, so in this particular experiment, there's about a 30% reduction in feed intake. And now all the data I'm going to show you for the rest of the 45 minutes here is not confounded by differences in feed intake. They're the direct result of heat stress. So here's the pair-fed cows. Remember, the blue line is the thermoneutral cows consuming 30% less feed. There was a reduction in feed intake of about 5 kilos, right? Because they're eating less feed. But now, only after about the first 24 or 48 hours, these cows will implement homeoretic strategies to maximize milk yield, right? They're here to make milk, and they'll do everything in their power to do so. The heat stress cow, there's just a continuous and progressive decrease in milk yield for about 6 to 7 days until it finally plateaus. So here's what we understand from this simple experiment is that all the area between these two lines has everything to do with just simply being heat stress, right? And by doing some math, we know about 50% of the reduction in milk yield during a heat wave is due to a reduction in feed intake. The other 50%, though, at this stage of the presentation, we don't know what explains that. What's causing this other 50%? And we've essentially spent the last 15 years of our lives chasing this down. So as the name implies, heat stress, right? Stress is associated with the stress response. You have an increase in CRF and ACTH, and eventually an increase in release of cortisol. And this is a dairy cow experiment where we saw about almost a 60% increase in cortisol. And of course, cortisol is a very catabolic hormone, right? It's responsible for breaking down different tissues. And not surprisingly, then animals both animals, the pair-fed, thermoneutral, and the heat stress animals, lose a considerable amount of body weight in that week or 9 days of heat stress. Right? Almost 100 pounds of body weight loss in a week. So this is a hypercatabolic condition. And as you'd anticipate, when animals are in thermoneutral conditions and consume 30% less feed and lose almost 100 pounds of body weight, their non-esterified fatty acid concentrations go up. So here's the NEFAs on the y-axis, and of course, non-esterified fatty acids are a product of adipose tissue mobilization. The pair-fed cows will mobilize adipose tissue in an effort to spare glucose for the synthesis of milk. So this is normal. This is 100% anticipated. The heat stress cows do not. And it turns out that there's a variety of models where heat stress animals just simply fail to mobilize adipose tissue despite the endocrine profile suggesting or predicting that they should, increasing cortisol, epinephrine, glucagon, etc. So these animals should be breaking down adipose tissue, but they don't. And again, this is conserved amongst a variety of species: ruminants, pigs, chicken, rodents, humans. Something is preventing adipose tissue mobilization during heat stress. Well, what's the most obvious explanation? It's insulin. Strangely enough, despite the fact that animals go into negative energy balance, they lose body weight, and there's this hypercatabolic endocrine profile, there's also the strange increase in insulin. I want to emphasize that again. This is weird because the animal's gone off feed, right? 30–40% reduction in feed intake. What's explaining this increase in insulin in dairy cows? Well, it turns out it happens in a variety of species. In fact, I think it turns out it increases in every species ever evaluated: beef, pigs, rodents, rabbits, humans, hell, even snakes. When they become heat stressed, there's an increase in circulating insulin, both basal and stimulated insulin response stimulated by a glucose tolerance test, the GTT. So this is a highly conserved response, and it's just really metabolically strange. Okay. So the nice thing about dairy cows, of course, is that we can do an accounting of glucose every time we milk them. So we know that the pair-fed cows, or the heat-stressed cows, are secreting approximately 400 grams less lactose per day than the pair-fed controls. In other words, there's almost a pound. Remember, there's 454 grams of glucose in a pound, and the heat-stressed cow is secreting about a pound less of carbohydrates less than the pair-fed thermoneutral controls. So the question we had next was, okay, is the liver producing 400 grams less of glucose per day? Or is some other extramammary tissue utilizing more glucose per day? So we utilized, we collaborated with a friend, Matt Waldron, and looked at using stable isotopes, looking at hepatic whole body glucose production. And I tend to get bogged down in the slides, so I'm not going to do that. But the main thing here is that the pair-fed and the heat-stressed cows have a decrease in their capacity to make glucose compared to thermoneutral ad libitum conditions. But the important thing here is the decrease in glucose production is similar between the two environments. So what does that tell us? It tells us that it's not the liver's fault. The liver's making glucose. It's responding to the gluconeogenic hormones. And importantly, the hyperinsulinemia is not shutting down the hepatic glucose production. So we discovered this back 10, 12 years ago now. And so for a long time, we assumed that the muscle was increasing its glucose consumption. And I'm going to try to convince you now that probably the increased utilizer of glucose is the immune system. And to get an idea of why that there's an immune response during heat stress, you got to understand how heat stress affects the gut. Okay, so every animal, when they start to accumulate heat, including humans, vasodilates at the skin. That's why when you go to the beach or you go to the gym or you bale hay or whatever, when you start to become hot, your face gets red, your skin gets red. You're doing that because you're trying to vasodilate and to maximize heat dissipation. Okay, but if you're going to have vasodilation at such a large tissue like the skin, you have to vasoconstrict somewhere else. If you didn't, you'd die from low blood pressure, not getting enough blood back to the heart. And the area of the body that vasoconstricts during heat stress is the gut. So you have this massive vasodilation at the periphery of the skin, and then an equivalent amount of vasoconstriction going on at the gut. And you'll see then here in a second why there's multiple problems with that. And it turns out the epithelium of the gut is very sensitive to reduced oxygen delivery or hypoxia. A picture's worth 1,000 words. Now this is the ileum of a heat-stressed pigs, but this is the thermoneutral ad libitum group where the ileum are long and slender. So this is a hallmark of a healthy intestinal barrier integrity. And the ileum of the heat-stressed, the villi are even hard to distinguish, so the damage is clear. Now, interestingly, from just simply having a reduction in feed intake of only about 40% damages the gut. The villi becomes shortened and fatter. Okay, so I don't have time today to talk about how off-feed events or inadequate feed intake can damage the gut as well, but it does. So remember, I talked about trillions of microbes in the gut, and if they get in through the barrier of the gut, they'll stimulate the immune system. Well, one of the things that's in the gut is called lipopolysaccharide, and in a normal human, there's well over a gram of it in the gut. So there's probably many grams of it in the gut of a cow. And in LPS, it's made by gram-negative bacteria. It's the outer membrane component of gram-negative bacteria. If it infiltrates a barrier like a scratch in your hand or your gut, it stimulates an immune response. And probably everyone on today's talk is familiar with being sick, right? You have reduced appetite, fever, muscle breakdown, you become lethargic, you get tired. So LPS is what we use in the laboratory to stimulate an immune response or try to model an immune response. Here's an oversimplified cartoon of what your gut looks like. These are two enterocytes, and the enterocytes create and synthesize these proteins called tight junction proteins. I read where there's at least 50 different types of tight junction proteins. Now they're made obviously within the cell, but they're embedded on the lateral side. And they reach out and they make a physical connection with their neighboring cells, tight junction proteins, and create almost like a Velcro barrier, almost like a zipper, for example. Essentially what it's doing is it's a gate in this paracellular pathway that prevents LPS pathogens and other antigens that are naturally in the lumen of the gut from getting in. Okay, but first, as it turns out, now we're learning more and more that any type of stress, even psychological stress, will cause these tight junction proteins to be pulled back into the cells that created them. They're still there, they're just not where they need to be. Now you no longer have this physical barrier, and you get the infiltration of LPS and undoubtedly hundreds, if not thousands, of different types of antigens will get in. And remember, most of the animal's immune system resides in the gut. So if the local immune system does a good job, you won't even know that you've had a leaky gut or you've had an infiltrating antigen. If the local immune system is unable to clear it, it travels via the portal blood to the liver. And the macrophages, the resident macrophages in the liver, they're called Kupffer cells, will hopefully do a good job of detoxifying that arriving LPS. If the liver is unable to handle all the LPS that it's arriving, now the endotoxin goes systemic, and the animal starts experiencing endotoxemia. Hell, it can even experience bacteremia. So what that means is that entire bacteria can seep through this paracellular pathway, travel through the portal blood, and get through the liver and go systemic. Okay, so the effects of heat stress on the gut are very quick. This is jugular vein LPS. So on the x-axis is time. And within 2 hours, you see this noticeable increase in circulating jugular vein LPS. The reason why I'm emphasizing jugular vein is because obviously, if you can pick up an increase in LPS in the jugular vein, the local immune response of the gut has been overwhelmed, and so has the liver. Now, these animals are experiencing endotoxemia. And then not surprisingly, because you have this infiltration of antigens, you're having an immune response. These are two acute phase proteins, LPS binding protein and serum amyloid A, and they have the heat stress on the x-axis. So when there's heat stress, you get leaky gut, you have infiltration of antigens like LPS, and you end up getting an immune response. Okay, pretty substantial immune response. So at this stage of the talk, the summary is that reduced feed intake only explains about 50% of the decrease in milk yield. There's a strange hyperinsulinemia. The liver's not responsible for the problem. It's making the glucose. The glucose is just simply not ending up in milk. There's leaky gut. There's an immune response. And in reality, the biological consequences of heat stress aren't much different than normal infection. Okay, and at this stage of the talk, we can't identify where this 400 grams of glucose is gone. So the gut becomes leaky. right? And there's a variety of different models that demonstrate that. And you're probably asking yourself, okay, what's the big deal? What are the consequences of this, right? So to answer that, we need to have a good feeling of what's going on with insulin. Why is there this strange increase in circulating insulin during heat stress? And it happens, incidentally, this also happens during infection, sepsis, bacteremia, and we can very nicely model this increase in insulin by infusing LPS. So what would be insulin's role during heat stress? Well, a good friend of mine, when he's working with Tom Overton and Yves Beauclair, was studying the consequences of metabolic consequences of mastitis. This is Matt Waldron's work from his postdoc. So they infused LPS up the teat canal at time 0. The cow gets a fever, completely stops eating, and approximately 2 hours later, there's this insulin surge, which is strange, right? The animal's got mastitis, severe mastitis. It's completely stopped eating. Why would the most anabolic hormone go up? Again, just a quick reminder how important glucose is. It takes 72 grams of glucose to make 1 kilogram of milk. And if anything reprioritizes the hierarchy of how glucose is used, it's going to cost, or has the potential to cost, milk. So that leads us to a guy named Otto Warburg. Otto Warburg was a professor in Germany, and he receives the 1931 Nobel Prize for identifying a couple of things. One is that cancer cells prior to becoming cancerous can burn a variety of different fuels. But once they become cancerous, they initiate this thing where they switch from oxidative phosphorylation, meaning they stop using the Krebs cycle, and they start generating their energy only from aerobic glycolysis, which is an inefficient system. He also recognizes that immune cells are the same way. So immune cells, before becoming activated by an antigen, are quite metabolically flexible. They can burn a variety of different fuels. But once activated by an antigen, their preference becomes glucose, and again, not through oxidative phosphorylation, but through aerobic glycolysis. And I'll show you a cartoon of that in a second. And so he wins the 1931 Nobel Prize. 3 of his former PhD students go on to win Nobel Prizes. One of them was Hans Krebs, and he was also good friends with Albert Einstein. So this is an amazing man whose influence on science is going on over a century now. So the Warburg effect essentially just demonstrates that you have a resting immune cell could burn a variety of different fuels. Here it's shown glucose coming in, going through glycolysis, 2 pyruvate molecules coming down to the TCA cycle or the Krebs cycle, and generating efficient quantities of ATP. This is an efficient process, but a slow, lumbering process. Now, an activated immune cell, activated, for example, by LPS, has a very rapid increase in need of energy. And so it no longer has the flexibility of burning different fuels. It only wants glucose. And of course, it needs insulin to get this rapid increase in glucose or uptake of glucose. So this is not anaerobic glycolysis like we all had to memorize in biochemistry. This is an aerobic glycolysis that is induced by an immune cell to generate rapid quantities of ATP. The net result then is this carbon is exported from the immune cell as lactate. And this explains then why an animal that's sick or has in some type of infection, it oftentimes has high levels of circulating lactate. Okay, so how much, right? How much glucose? Remember, it takes 72 grams of glucose to make 1 kilogram of milk. Well, this has been difficult because it's very hard to isolate and localize the immune system. Every tissue has resident immune, even the brain has resident immune cells. And to make matters worse, it migrates, right? If you got an infection in your big toe, your immune system will migrate to the big toe. So you can't isolate it. So attempting to quantify the glucose needs of an immune system requires you to do that at the organismal level. So we initiated this thing called an LPS euglycemic clamp, and it's a fancy word for essentially just infusing glucose. we give glucose at, or sorry, we give LPS at time 0, there's a couple hours of hyperglycemia. And this hyperglycemia is because the liver's glucose output increases. Muscle and adipose tissue stop using glucose, peripheral insulin resistance. And these three systems outweigh immune system glucose utilization. But once the immune system gets fully engaged, and almost all immune cells do this, by the way, almost all leukocytes, now their glucose utilization will outweigh the body's strategy to provide glucose for the immune system, and they become hypoglycemic. Okay, so we kind of thought, well, what if we can start infusing glucose the second she becomes hypoglycemic? Could we get a proxy of overall glucose use? So graduate student injects LPS into the jugular vein. We take blood samples, we measure the blood glucose levels, and as soon as she becomes hypoglycemic, we start infusing glucose. We do this for 12 hours. We subtract off the controls, and it's approximately 1,000 grams of glucose, a kilogram of glucose in a 12-hour time frame, extrapolated to a day. We're talking 2 kilos. 2 kilograms of glucose is an enormous amount of energy, right? Here's a 1.81 kilograms of table sugar. And so this is an enormous amount of utilization of energy, and we've done this now in a variety of different species. And when you place the data on a metabolic body weight basis, I don't think it's surprising how similar then these data are. About approximately a gram of glucose per kilogram of metabolic body weight per hour. So remember back earlier when I said we had heat stress cows were secreting about 400 grams less carbohydrates per day than the pair-fed controls. Well, right, the cow gets heat stress, it gets leaky gut. Leaky gut stimulates the immune system. The immune system now is the #1 priority, and the immune system starts to utilize glucose, and the mammary gland is just no longer high in the totem pole. Okay, so what can we do about it? Right? What can the feed and animal health industry do to fix leaky gut and heat stress? Well, there are a variety of different targets, increasing feed intake, direct actions at the intestine, and we'll talk more about some of these dietary strategies here in a second. But by far the biggest impact a producer can do is heat stress abatement, right? Physically modifying the environment is by far, I can't emphasize that enough, the best investment of money. And I've just kind of listed them here: shade, fans, soakers, and misters. And so what are the benefits from investing in cooling? Well, I think I would assume most people have a fairly good feel for this already, but obviously for improved production, efficiency, and profitability. Summer fertility is a huge issue. Probably 50% of the decrease in the economic consequences of heat stress are poor reproduction, welfare, health, sustainability, right? And so what would the priorities of heat stress be? Well, I'll show you here in the next slide, but solar radiation is incredibly intensive. I'll have to show you some data to prove that. I think most people know that. And the second then would be to utilize evaporative cooling, of course, while incorporating fans. So this is very old data now, right? 70 years. But this is just simply looking at black dirt surface temperature. This is data from Nebraska. Black dirt surface temperature during the middle of a July day. So in the sun, no shade. At 4 o'clock in the afternoon, the black dirt temperature was 153 degrees. And just simply providing 5 minutes of shade will markedly reduce the temperature of that ground. So the emphasis here I'm just trying to show you is the power of shade, right? I think everyone probably has familiarity with that already, but just simply providing a small amount of shade can markedly reduce the thermal load on an animal. The other one I just want to talk about quickly is evaporation. And I don't want to freak you out and remind you about physics, but the amount of energy needed to evaporate water is enormous, right? 540 calories to evaporate a gram of water going from 100 degrees Celsius into steam. Well, this explains why steam burns are so dangerous, right? Because there's so much more energy in the steam than there is in the hot water. And so let's, well, incidentally, there's also energy liberated during condensation. So the same thing is true going both directions. One, evaporating or vaporizing water. The other one is condensation. And we'll give you examples of that both here in a second. But so the evaporation is a cooling process. I've already talked about the quantity of energy needed to evaporate water. And this explains then a variety of things that we all have familiarity with, right? When you get out of a swimming pool, it might be 100 degrees and you still get chilly. Or when you get out of your shower, before you got in your shower, you weren't cold. But now when you're coming out of the shower, you're cold. Well, why is that? It's because when you're wet and you're walking out now of the pool or your shower, the water that's on your skin evaporates. And the energy required to evaporate that water is coming from the thermal energy on your body or in your body. So the water evaporates, the energy required to make it evaporate is coming out of you. So that evaporating water is literally sucking energy, thermal energy out of your body. Okay. This also then explains why it's warmer to stay in the shower, right? You get out of the shower, you shut off the shower. Why is it warm inside the shower? Well, because that water that was vaporizing during your shower is now starting to condense. And condensing liberates heat, right? So we talked about the opposite, right? Condensing water liberates heat. That's why it stays warm inside your shower, even after you shut off the water. And when you get out, that water's evaporating off your skin. That's why you get cold. Others examples are, for example, blowing on hot coffee. What you're doing when you're blowing on hot coffee is changing the gradient of the temperature, allowing that the energy then to vaporize that water is coming from the thermal energy in the coffee. So when you blow on water and you see the steam, or sorry, hot coffee, and you see the steam or the vapor leaving the coffee, the energy required to vaporize that is coming from the thermal energy of the coffee, and the temperature of the coffee then goes down. I'm emphasizing this, everyone, because I think everyone has familiarity with the power of evaporative cooling. Sometimes you just don't wrap your head around or think about it. So there's three ways to utilize evaporative cooling is to cool the cow. In this particular strategy, you're literally putting water on the cow's skin and evaporating it off, right? And again, the evaporation of that water on the surface of the cow, the energy required for that is coming from the body temperature of the cow, and the cow becomes cool. Cooling the air, this is a strategy that's oftentimes utilized in more arid climates like Arizona, right, where the producers there will introduce very small droplets of water around the cow. That water will evaporate, hopefully before it hits the ground. And again, the energy required to evaporate that droplet of water is coming from the air temperature in this particular example. And now the air temperature has gone down because the energy has gone into evaporating that or vaporizing that water, or some type of combination of the two. I got this slide from John Smith, the late John Smith gave me this slide, and a couple of the others. Priorities to reduce heat stress. Obviously, cows will increase their water consumption when hot. To provide shade, we talked about why that is. And importantly, don't forget about the dry cows. I'll show you some data here in a second about dry cow cooling. Utilize evaporative cooling. And even in relatively humid climates like Iowa and the Northeast and Northwest, evaporative cooling remains effective. Not clearly as effective as in more arid climates or low humidity climates, but it remains effective. Fans can utilize to to dissipate heat and also to increase evaporative cooling. Minimizing distance to the parlor. Holding pen is almost always the spot on dairies where cows get the hottest. Improve ventilation. And where to do it? You know, long story short, if you can afford it, everywhere, right? If you can. So the summary of heat stress abatement, providing good shade, utilizing evaporative cooling, more water the better. And the frustration I get from, I can hear the frustration oftentimes from producers. Well, I have to get rid of this water. Now, in part of this evaporative cooling is utilizing, is managing the evaporative cooling to try to minimize wasting water. But, and again, also I don't know, what the cost of water is in everyone's systems. But don't forget about the dry cows. I'll show you data here in a second. And try to measure it. If you're going to invest in cooling, try to get some metrics that allow you to recognize that your investment's paying off. So this is some data from Dennis Armstrong, who was on faculty at University of Arizona when I first got there, and he's a legend in heat stress abatement strategies. He was working with dairy farms in Saudi Arabia. And so three different dairy farms over here in the far left column, and they were heat stressing. They were cooling dry cows or not. That's the only treatment. Okay, so in the dry period, cows are either cooled with shade and evaporative cooling, or they weren't. And then once they calved, all cows were cooled. And these are just different metrics of reproduction. And in all metrics and in all three dairies, the cows that were cooled pre-calving had improved metrics of reproduction. And on the far right, I'm sorry, is culling because of reproductive purposes. So clearly the benefits of cooling a dry cow extend far into lactation. Bob Collier and others were also looking at the biology of this at the University of Florida. And Jeff Dahl and those guys are also extending that. So there are marked improvements, future improvements. We're talking 60 to 100 days following that cooling on reproduction. So, I'm a Star Wars guy, but I just hope now everyone has a good feeling for just how powerful evaporative cooling can be. Even in climates that are relatively humid, evaporative cooling, if managed correctly, is still a very good use of invested money. Some management strategies. I already talked about reducing the distance from the parlor. The holding pen is where cows get packed in like sardines. They become stressed. Consider exit lane cooling. I think that's a good strategy. Try not to lock up during the middle of the day. A good friend of mine, short feeds. So if you can know when a heat wave is coming, short the cows about 5% of feed on the day before, and then on the day of heat wave, they're aggressively eating. That also requires quite a bit of management. Feeding later in the night, early in the morning, push up feed often. If you put your hand in the feed and it's hot, they're not going to eat it. And I understand the producer's reluctance to throw it away, but hot, moldy feed, fermenting feed they're not going to eat it. Try to avoid vaccinations during the middle of the day. Of course, vaccinations are accompanied by a small field oil response, and at least provide shades for the dry cows, if not some evaporative cooling. So dietary strategies, right? These are mostly, I think, theoretical. I have a hard time studying these at the university, but low heat increment diets, trying to save your best forage for the warm summer months. Fat, there's surprisingly not as much research on dietary fat as you'd think, but most of them do show some type of improvement of increasing fat content of the diet. And of course, that's because the digesting absorption of fat is accompanied with low amounts of heat, a balanced amino acid profile, with regards to supplements, I think there's a variety of things and different strategies, right? I haven't even talked about how heat stress causes rumen acidosis, but modifying rumen environment, supplements aimed at improving gut integrity, feed intake stimulation, immune modulation. The one I want to talk about today is vasodilators. Okay, when I was at Arizona, Bob Collier was leading an effort looking at how niacin affected heat stress in dairy cows, and his PhD student was Rosemary Zimbelman. And so the first experiment they looked at, I think this was 12 grams of niacin a day, and this is vaginal temperatures of heat stress cows when they're, and the controls are in black, and when they were fed the niacin, it was in the open gray, I don't know how these colors. Anyway, these are the animals that were fed the niacin. So there was a substantial decrease in body temperature when they were fed the niacin. And Bob did another experiment looking at a dose response. That particular experiment didn't reproduce the body temperature data, but they did see a linear increase in water consumption with increasing doses of niacin. And then Bob did another experiment looking at the molecular consequences of betaine and niacin. And interestingly enough, and I'm not going to get through all the heat shock proteins in all the nuts and bolts of Bob's paper, but what I'm trying to demonstrate is that the effects of niacin on the cellular machinery that regulate the heat shock response are pretty significant. Okay. So back to the gut, I think leaky gut can explain a variety of things. And again, I didn't have time to talk about ketosis and off-feed events and fatty liver, but I think there's a variety of things in animal agriculture associated with leaky gut. And in large part, it's just due to stress. So the stress umbrella that we've been developing in our group, where you have these different stressors and causing reduced feed intake. And this reduced feed intake is just only part of the problem. The other part is this leaky gut induced immune activation, and then nutrients being utilized that would otherwise have gone to making of milk or a fetus or muscle. Okay. So nearing the end here, heat stress cost it decreases almost every metric of productivity and profitability. And it's not just a large or small dairy farmer issue. It costs everyone in the industry. Reduced feed intake is only a small portion of the problem. There's this heat-induced leaky gut, and essentially the biological consequences of heat stress are incredibly similar to just simply other immune activations, mastitis, meningitis, etc. From a dairy producer's perspective, heat stress abatement is by far should be their biggest priority. And once they've invested in then these infrastructural changes, dietary strategies, I think, are also a good strategy to minimize the negative consequences of heat stress. The USDA has been very good to us, and this is my team. I have a team of rock stars, and they do all the work and collaborators. So I hope you found the hour here useful, and I'm happy to take any questions if you have them.
Tonight's last call question is brought to you by NiaSure Precision Release Niacin. Niacin is a proven vasodilator for heat stress reduction and a powerful antilipolytic agent for lowering high blood NEFA in transition cows. Protected with Balchem's proprietary encapsulation technology, you can be sure it is being delivered where and when your cows need it. Learn more at balchem.com/NiaSure.
Dr. Baumgard, our first question is, if you were implementing a cooling system on a dairy and you could only do one project at a time, where would you put your first priority?
Yeah, so I'm gonna, I've heard Bob Collier and John Smith been asked this question many times, and I'm just going to essentially regurgitate what I've heard them respond. First is making sure there's plenty of water, access to clean water. And this is probably the cheapest thing you could do, is just throw the grandkids out and make sure you clean the water tanks, right? Second one would be shade, preventing that solar radiation. And then the third is utilizing evaporative cooling with fans.
All right, excellent. Next question. With regard to heat stress and LPS, how significant does a bout of heat stress need to be to initiate the response? For example, if a group of cows experience heat stress during the day, but it cools off at night, will that initiate leaky gut, or does it require a more prolonged exposure?
Yeah, that's a great question. And I'm gonna allow productivity to help guide my response here. If a cow is able to cool off that night and eliminate all the heat that she was accumulating during the day, the effects on milk yield seem to be pretty small. And all you need to do is go to visit some dairy farms in New Mexico versus Arizona, and look at the infrastructure and the amount of money invested in the different dairies to know that, right? And so it cools down much better in New Mexico than it does Arizona. So yeah, the question's hard to answer. How many hours of accumulated heat is required? Because of course you have cows that are acclimated and not, blah blah blah. But if they're able to get rid of that heat and start the next day with their body temperature back to normal, they seem to really tolerate heat stress pretty well. So the amount of heat required to cause this leaky gut must be some type of 24 hours, 36 hours, but it's a good question.
All right, very well. Let me see if I can get the gist of this next question. Is there a level of leaky gut? Question mark. For instance, a level where only toxin can access the bloodstream and a level where bacteria access the bloodstream?
Yeah, that's a great question. And I suspect there is a continuum, right? And where the early stages of impaired intestinal integrity, you probably get the small products of bacteria like LPS and other antigens. And then as the gut becomes more severely injured and the paracellular pathways become even more and more open, then you'll get larger molecules like entire bacteria getting in. It's a great question. I'm unable to give you a specific time or heat load, but I suspect there's that continuum where it's first LPS, endotoxin, and then eventually whole bacteria.
All right, thank you. Next question, where do you think the gut gets most of its energy from? Oxidation of VFAs? If so, can we feed the rumen better to potentially preserve gut function?
Yeah, great question. And I think there's of course, butyrate is a key energetic substrate for the rumen and for large intestine. Butyrate's contribution as an energetic fuel source for the small intestine, I think, would probably be less than these other two segments. Glutamine, and glucose are also sources of energy for the small intestine. I think there's strategies at trying to maximize butyrate's contribution to energetics in the large intestine. It's a hard question, and you're probably getting tired of me saying I don't know. How to feed the rumen to maximize butyrate's contribution to the lower gut. Encapsulating it or providing a precursor to butyrate that reaches the large and small intestine, I think there's potential value there. It's a good question.
All right, I think we've got time here for a couple more. Lance, next question is valuable information regarding energy metabolism in heat stress animals. As a primary nutrient, amino acids metabolism also can be changed in heat-stressed animals. Do you have any data regarding the amino acids composition changes in the physiological status of heat-stressed animals?
No, not really. I'm going to extrapolate from other species. So the immune system has a huge increase in glucose metabolism use, but it also has a huge increase in amino acid requirement as well. And the issue with amino acid metabolism during heat stress is that, so I didn't talk about the mobilization of skeletal muscle during an immune response, but they do. Animals will mobilize skeletal muscle. But the amino acid profile of skeletal muscle is different than that of the acute phase proteins. And that's one of the big reasons why the skeletal muscle is being mobilized, to provide amino acids to the liver, for the liver to utilize those amino acids to make acute phase proteins. But the amino acid profile is different between the two. So skeletal muscle is mobilized in excess of what's needed to make the acute phase protein. That's why blood urea nitrogen markedly goes up. So I think there's opportunities for us as nutritionists to get a better understanding and be able to predict what that amino acid profile is, and then thus feed to meet those requirements. And that's not just a heat stress thing. I think it's also a transition period issue as well.
All right, I'm mindful of our time here, and I think we have time for one more question. I do want to tell everyone that if your question was not answered today, we will be forwarding these to Dr. Baumgard, and he will answer them, and then we'll make them available on our website, balchemnh.com/real-science. So last question, how many days/weeks before the first heat wave arrives should we start feeding additives to help heat dissipation?
Well, I think you definitely want, from a nutrition perspective, to preemptively get there before the heat wave gets there. So if you're able to, it depends upon which supplement you're talking about, probably. And the Balchem scientists probably have a better idea of how long niacin needs to be fed before you get the vasodilation effect. Depending upon the supplement, if it's a gut issue, I would think you want to have it preloaded before the heat arrives. Oh, yeah, I don't know. A week, 5 days. The problem is accurately predicting heat waves is difficult unless you live in Arizona where it's hot all summer, Texas, Florida. But in the Midwest and Northeast, of course, we have heat waves. So it also depends upon the cost of the supplement and everything. So I'm not able to give you a good answer. In fact, I don't think I've answered a single question yet. I apologize for that.
You've been awesome today, Dr. Baumgard, and I want to thank you, and I want to thank everybody for attending today's webinar. On behalf of Balchem and Dr. Baumgard, thank you for joining us today.
We'd love to hear your comments or ideas for topics and guests. So please reach out via email to anh.marketing@balchem.com with any suggestions, and we'll work hard to add them to the schedule. Don't forget to leave a 5-star rating on your way out. You can request your Real Science Exchange t-shirt in just a few easy steps. Just like or subscribe to the Real Science Exchange and send us a screenshot along with your address and t-shirt size to anh.marketing@balchem.com. Balchem's Real Science Lecture Series of webinars continues with ruminant-focused topics on the first Tuesday of every month, monogastric-focused topics on the second Tuesday of each month, and quarterly topics for the companion animal segment. Visit balchem.com/realscience to see the latest schedule and to register for upcoming webinars.